at the Institute for Cross-Disciplinary Physics and Complex Systems at University of the Balearic Islands. After that, in 2014, he was a postdoctoral researcher at the Department of Ecology and Evolutionary Biology in Princeton. And since last year, he's a Simons professor at ICTP and CIFAR and IFT. And he's also a Simons Associate at the Quantitative Life Sciences at Abdus Salam ICTP in Trieste. And his main projects are involving microbial ecology and evolution, animal movement and search theory, pattern formation in ecological systems, and stochastic models of population dynamics. So right now I'm going to give the word to Professor Ricardo, and I will let you know when you have like 10 minutes left, okay?
Okay, thank you very much, Raul. Thank you very much for a nice introduction. So, well, before I start with the presentation, I would like to thank you for the invitation. It's great to talk here. The second year I'm in Brazil and seeing so many things going on. It's very exciting. And also congratulations for the great job you have done organizing this huge conference online and in these times. It's not easy. So congratulations and thank you very much for running these events that are so important for everyone. So yeah, this being said, I think I'm going to start sharing my screen and we can go to the presentation. So yes, I think now you can see my slides, right? Okay, so the main goal that I fixed when I started thinking about this presentation was to provide kind of a broad overview of the things that we are working on and the approach that we are taking and how we understand the science that we do. And I thought that the best way to do that is by addressing the question that I actually put in the title of my presentation, which is how can statistical mechanics or statistical physics, which is a branch of physics that we as physicists are very familiar with, or we think we are familiar with, and how can we use that to help other fields? And in particular, I'm going to focus on population ecology because that's the where most of the questions that we address lie on. But the approach that I want to take for this presentation is an approach that is actually also very related to the approach that we take in the work that we do. And it's not only thinking how statistical mechanics can help ecology, but also how can ecology help statistical physics and physics in general. So how can this relationship be a two-way relationship? So both of them are benefiting from each other. And to start discussing this question, I want to draw a very brief picture of how these disciplines that in principle are so different, right? How does all these statistical physics concepts that we know, how do they relate with population ecology, which basically relates to birds and animals and bacteria? And although they may seem very, very different, at the end they have some principles that are actually very similar. And that's what I want to bring up to begin with. So statistical mechanics in principle is this branch of physics that tries to connect the microscopic properties of inanimate matter most of the times, or originally, with the macroscopic properties that we can measure. For instance, in the case of a gas, we try to relate the mean velocity of the particles, or the mean kinetic energy with the temperature of the gas. That's a classical problem in statistical mechanics. So at the very, very core of these problems is this connection between the microscopic scale and the macroscopic scale. And that's something I want you to have in mind for the rest of the presentation. So what happens when we turn to population ecology? Well, population ecology, the first thing that we see is that the systems that we are working with are in principle prettier, or at least it seems to me that a tree with birds is prettier than a box with a gas. It might be not a universal truth, but it seems to. And the goal of population ecology is actually to understand how this number of birds changes in this tree. Of course, I'm oversimplifying, but the kind of questions is to understand how these population sizes, how they change in numbers and how they change in space and how they change through time, basically. And the processes that drive these changes are basically are very different, and they may be very, very complicated. But if we want to take a simplified picture, we can think that, well, I mean, birds increase, the number of birds increases when there is a new guy that is born, but it can also increase when new birds arrive to the tree because they think it's a very nice one, or because they have better conditions to live. But it also decreases, right? We have these very large eagles that are going to eventually eat our tiny birds. And they may also, other friends of the birds that were eaten, they can see this and they can decide to leave. So we have very different processes that are going to change how this population and how many birds we have in this tree. So this picture that we are drawing in this way, what it's actually telling us is that we have in our population, we are going to have very different processes that actually involve individuals that we are only going to be able to see through the change in the microscopic scale. Of course, you may think here, well, I mean, I can watch the tree forever and I can see when birds are born and when they leave. I can count them one by one, which I cannot do with the gas because the number of degrees of freedom is very different. And you are totally right. I mean, this is just an example. We'll have systems in which this cannot be seen, okay? So in principle, the idea is that we have also microscopic processes, in this case, birth, death, immigration, emigration, that at the end are going to result on a microscopic feature of the system, the number of birds. That is what we have access to. Okay, in this case, it's not such a big black box, but this mapping is kind of there. So with this in mind, the statement that I always like to make when I start presenting this approach is that these population dynamics, these changes are, it is an emergent phenomenon indeed. It's not, I mean, it's something that relays on very important and many times complex microscopic processes. So to illustrate that, I want you to think a little bit about an experiment that was actually done now almost 100 years ago by this guy, by Gauss. And the experiment is very, very simple. So he basically took some yeast. Yeast is a microorganism. It's very, very tiny living beings that do very important functions in our daily lives. For instance, they are responsible of bread, and they are responsible of beer, wine, and all these important things for human beings. So he took this yeast, he put them in a bottle with a lot of food, and he allowed them to grow for many time, for a long time. And he basically developed a way to measure how many cells he had in that bottle. And what he found is that when he was representing this number of cells as a function of time, this was kind of the behavior that he saw. He saw that first the number of cells grows, and eventually it stops doing so. Then he repeated the experiment with different yeast. That here I'm going to represent it by red, but in principle, I mean, in being more precise, it's a different species with a different name. And he repeated the same experiment, and he did the same, and he obtained this other pattern that you can see here. So he basically, I mean, the curve is in principle the same, at least qualitatively, but please notice that the axis, the scales are very different. The time scale of the growth is much larger in this case, in the red case, and also the number of cells that are going to grow is much smaller. So we have this idea, we have this concept of the pattern, the macroscopic pattern having some qualitative common features, regardless of the specific cells that I have, how big they are, how fast they eat. All these details in principle are not important if I want to take a qualitative shape of the curve, but they will be important for other questions, of course. So this kind of generality in these behaviors started attracting the attention of many people, many mathematicians and physicists. And actually these people had been thinking in these problems before this. When they were thinking about how these patterns can be explained, they started trying to develop mathematical models that were as simple as possible to be tractable, but still general to reproduce this behavior. And this equation that you have here in the slide, which is actually, I have to say, one of the very few that you will see, I'm not going to use a lot of math, is what is called the logistic equation, which is the most well-known model in population dynamics. So this equation basically gives us the rate of change of the population size, that here I'm calling it X, but you can use, of course, wherever. And this equation is telling us that this change has two contributions. It has a positive contribution that is proportional to the population size, and it has a negative contribution that is proportional to the square of the population size. So basically, when X is very small, this term is going to dominate and my population is going to grow. But when X is larger, then this square term is going to dominate and it's going to stop the growth of the population. So if we go back to our experiments, we can try to fit the solution of this model to our data. And as you can see in the slides, the agreement is pretty good. Of course, the parameters of the fitting are going to change depending on the species. But again, the qualitative behavior of the population is reproduced by this dynamical system, by this dynamical process, which is the key ingredient of all the models that you can see in population ecology. Right, so up to this point, everything is very simple. We have a ODE that for us, for physicists, is not very challenging. We like multidimensional integrals. Many of you, I'm pretty sure you do. For biologists, it's great because it's simple. They can do it with the computer and they don't have to develop very strong mathematical skills. They have other skills. And everything is perfect so far. So the idea is this, no? We have a lot of cells that are in my bottle. They are all well mixed. And this is key. They are in a bottle that is containing water with some sugar or whatever, but it's water. So they are all over the place. This guy here has the same probability of finding this guy here than this one or this one because they are like an ideal gas. They are all over the place. And when I have these conditions, I have this macroscopic pattern that we already saw. But things are not as easy as we are drawing now. I mean, there is dynamics behind this pattern. There are microscopic processes that are happening there. In this case, we can write them using some kind of chemical notation. This is the typical notation that we use when we work with stochastic population dynamics that actually captures the distributedness of the population, right? So here what I want to say is that my cells, one of them is going to divide and produce two cells, and that's going to happen with a rate, which is a probability per unit time that I'm calling R. But my cells can also die because they are old or because they stop functioning or whatever. They die and they disappear is what this empty means. Or they can also it can also happen that because they are so crowded, they fight with each other. They collide and one of them kills the other. They struggle to survive. So this is actually what is happening at the level of the individual, not at the level of the population where I have this thing. But of course, if there is, I mean, if this is what I see, then this somehow has to lead to this. And here is what this, with actual mathematics and tools brought from statistical physics come into play. And all these techniques allow me to move from this description at the level of the individual to this description at the level of the population, which at the end I can tell you that is just the mean of the number of particles that will emerge from this stochastic dynamics. So basically, if I take these processes and I take some average, I can get this ordinary differential equation that gives me this microscopic. Okay, so everything up to here is very nice, very easy, and very interesting and easy to follow, I guess. But of course, this is a very reduced picture of nature, and this is not what is going to happen in many biological systems. And again, what I'm going to do from now, which is explaining the things that we do, is not, I mean, I don't want to, in any sense, I want to say that this approach is not useful. This has provided a lot of important knowledge, and it still is doing so. But I'm just going to make the point that sometimes it's not working, and studying the systems in which it is not working is our goal. And when we move to those scenarios in which this approach is not working, we find the question that is actually driving all the work we do at ICTP and all the work I've been doing for the last years, which is, okay, this simplified picture is very nice, but what happens when the complex microscopic features of this system translate to the macroscopic dynamics? So what happens in that case, and how do these complexities at the microscale are going to make my macroscopic models change? That's the main question that we work on. And we address that question in very different systems, from microbes, animal, plants. We use computation, we use math, and I'm going to discuss some of the cases that we work with, focusing on one, but I'm going to give a broad overview. So for instance, the one system in which we are working that is actually a project that is led by Vivian, who is a postdoc at ICTP, is to understand what's happening when this well-mixed case is not working anymore. So imagine that our cells, instead of living in a bottle that I can mix very nicely, they are living in the sea. They are living in an environment where the flows of water are not so nice. It's not mixing everything well. We have vortices, we have turbulence, we have chaotic mixing, we have advection. So how these external flows are going to change the dynamics of the population? This is the question that she's working on. And I can give you here some, I think I have to change, sorry, the laser. I can give you here some intuition, no? Here we are, sorry, what you're going to see is a bunch of cells that are going to be living in a flow, in one of these complex flows, and you're going to see how not well mixed they are, let's say. So let's let the movie go for a while. You see that they are already trapped in a vortice, and you see that now particles are trapped here. This is going to eventually break, and they are going to disperse to a different place. So this is not well mixed at all. There will be cells that will be closer to each other for some time, and then they will move away. So how is this going to impact my models is something that we are trying to understand. It's not a so well understood question, and it's something that Vivian is working on. So another example that you can find out there, which is actually one that I'm going to focus for a little bit more, it's the case of animals more broadly, animals that are living in the wild, large animals, mammals, birds, or any animal that we can see. And so, for instance, I'm going to show you here now a movie of some wolves that were tracked in Minnesota in the United States. And the way that we are able to create these movies is because there are ecologists, there are people that they go there and they put GPS into these animals. So it's kind of giving them an iPhone that is, or a cell phone, let's not make any brand advertisement, a cell phone that is going to send the signal of the animal to some satellite, and we are going to be able to record these trajectories. So this is for the case of wolves. Wolves in Minnesota, that they live in packs, they live in groups, and they are kind of together within groups, but they don't overlap. I mean, they don't like so much being with guys from other groups. So let's see what happens when we plot these trajectories. So here you will see that they start appearing. We have 6 guys. Each of them is given by a color. And you can see already that this is everything but well mixed, right? I mean, I have 6 very well-defined territories. I have magenta, green, yellow, red, blue, white. And they, for sure, they don't overlap at all. I mean, they have some overlap, but it's not well mixed. The white is very unlikely to find the magenta, and the blue, the red, and so on. So this is, for instance, a very clear case in which this assumption is absolutely wrong. So how can I use this equation to study this population if the very, very core of the approximation is wrong? And of course, to address that question, we will need to develop more sophisticated models, right? And here is where more developed techniques from statistical physics come into play. We were saying in the beginning that these well-mixed equations, this logistic growth, was assuming that individuals are an ideal gas. They are all over the place, well-mixed, they cross with each other, they do other things. So if it fails here, then we need to take a more sophisticated approach, and we as physicists, we know that, or at least me as a physicist, I know that I analytically can solve very few things. I know that I can analytically solve things if n is equal to 2. So, okay, I will have a lot of individuals, but I want to make my picture as simple as possible to begin with, and I'm going to assume that I have two individuals, okay? The two-valley problem. And the other thing that I know how to solve is the harmonic oscillator. So I'm going to see how far can I go by assuming that my individuals, instead of being an ideal gas that is all over the place, are particles that are trapped in these harmonic oscillators, and how far can I go from there, okay? So I will try to wrap up this problem, and then after I approach this question, how 2 individuals encounter with each other, then, okay, I will try to scale up to more individuals and different, more complex scenarios. But I want to keep it simple in the beginning. So I'm going to use these 2 assumptions. N is equal to 2, and my animals are going to be harmonic oscillators, classical harmonic oscillators. But let me explain this more carefully. What does it mean? So first, why is this assumption of considering n equal to 2 reasonable, and why is this insightful? Well, it is insightful because although at the end we have all this large population that is, some individuals are dying, some are growing, at the very end, what is actually driving that dynamics is the encounter between two individuals. So imagine that we have, let's say, two animals that are competing for some resources. Here we have a hyena and I always forget the name of this animal in English. Buitre. I think it's craven, no? Anyway, we have two guys that are fighting for some food. And if they encounter with each other, they will interact. But if they don't encounter, they won't. But we also have, for instance, mating, reproduction. I'm not going to explain here why encountering is important for reproduction in the case of animals. We also have other processes, predation. There is no way that this guy is going to kill this if they don't find each other. At least two have to find, right? Or more frequently in the media now, disease transmission. If two infected people don't find each other, or at least are close enough, the disease is not going to spread through a population. So we can in principle take n equal to 2 as a good thing to start seeing how things change. Kind of makes sense. And the other assumption that I told you I was going to make is that my animals are going to be trapped in a harmonic oscillator. So what does this mean? I mean, it's kind of sounds, at least to me, if I were not familiar with this, it would sound kind of weird. Well, the only thing that I'm meaning is that I'm going to use a well-known model, stochastic process, that actually is modeling the movement of a particle that is trapped in a harmonic oscillator and has some thermal fluctuations that are going to make it fluctuate within that potential. This is basically what this equation is telling me. This equation is telling me that C, which is the two-dimensional position of the animal, has the change of the position has two contributions. One is going to be a deterministic contribution that is going to drag the particle towards some position. This lambda is a parameter. You can think of your house, for instance. Your house is this center of the space that you occupy and would be represented by this lambda parameter. And then we have another contribution that is this stochastic term that is basically going to say, okay, my animals have some weird behavior that I cannot understand. I cannot resolve it, and I'm going to assume they are the result of some stochastic process. So what I can do is I can move from this description based on a stochastic differential equation that gives me the evolution of this Z, and I can move to a description that is going to give me the probability of finding my individual in a given position of the space. So if I don't have my stochastic term, if G is equal to 0, then this is an ODE that I can solve. This is an exponential ODE. It's an ODE whose solution is an exponential and tells me that C goes to lambda in the stationary state. It's not very complicated. But because I have that noise, now my description has to be probabilistic. My animal sometimes is going to be in lambda, but it's going to be dragging around, right? And that's what I can do. I can obtain that solution, that is the PDF, the probability density function. And it turns out that for this process, because I'm taking this noise with some statistical properties, this is a normal distribution. It's a two-dimensional Gaussian. And I can calculate the mean, which is going to be basically given by the coordinates of this lambda. And I can calculate the covariance matrix that is going to give me the width of the distribution and also the correlations in the distribution. I'm going to keep things simple, so the correlations, they won't exist. I'm parameterizing my model so I don't have correlations. And the only thing I have is some width in the axis that actually I'm taking it to be equal. So this is very, very simple. If we represent it, you can see that, for instance, I can run a numerical simulation of this process. I integrate my equation numerically, and I generate, for instance, this gray trajectory, right? There's no way, there's no way I can anticipate where the animal is going to be because this is stochastic, but I can actually calculate this PDF, and actually you can see this dashed line here, the thinner one, that is limiting the region of the space in which my animal is going to be with a 95% probability. So if basically what this is telling me is that if I leave my animal move with this model, but I don't know the model, my animal is moving around, and I take a picture of my animal, and I take 100 pictures of my animal, 95 of these pictures are going to be in 95 of those pictures, the animal is going to be inside this circle. Okay, so this is what my PDF is telling me. Perfect. So this is how I'm going to assume that my animals move. And this the radius of this circle is going to be proportional to these parameters of the model. Anyway, the important thing I want you to take from this slide is that my animal is going to have some kind of restriction in the use of the space. 95% of the times it's going to be here. And I'm going to call this region the home range. Is the home of the animal. Is the fraction of the space, the region of the space where my animal is more likely to be. Okay, so let's move on. Now we were saying that we wanted to compare this model, this more refined model, with the one that is basically at the core of the ideal gas. So the ideal gas, I think most of us know how it works. It basically assumes that I have a bunch of particles that they are inside a container. They are moving around and they don't change directions until they rebound with one of the borders of the container. You can also get to the same result if instead of assuming this ballistic motion, the particles are moving with Brownian motion and they rebound in the container. You get to the same conclusions. So as I was using before my stochastic differential equation, now I can do the same. I can write an equation for that dynamics. You can see already that I don't have that deterministic drift because I basically killed my potential. Now this particle is moving freely. But now, of course, if you are already familiar with the Brownian motion, this variance of the Brownian motion diverges with time, right? It's not going to reach a stationary regime as the other model did. So now I need to introduce boundary conditions in my model to actually trap the particle, to not allow it to move everywhere in the world. So now I have actually my range is going to be reflecting. So that's why this line is continuous instead of dashed. And if I simulate these dynamics, my particle is going to move all over the place and rebound and move and work. If I run this forever, at the end, all the container is going to be gray. The particle can be everywhere, which means that if I go to time going equal to infinity, the PDF, what before was a Gaussian, now is a uniform distribution. A particle is equally likely to be everywhere. And this is true if my container meets some smoothness conditions that here we are going to assume it does, okay? So let's summarize. This is what we have. We have on the right the typical models of Brownian motion in which basically my animals are going to be moving all over the place, rebounding, and the probability of finding my animal is the same everywhere in the place. On the other hand, I have these more refined models in which the animal, it doesn't matter if I define this large region, which is the same as this here, my animal is not going to use all the space and it's not going to use it in the same way because now I have a Gaussian distribution that is telling me that around here is more likely to find the animal than here or here or here. So very important difference. By adding this model, I'm introducing an additional spatial scale in my problem. Now, the important scale is not only this large R, but it's also this small rho that actually limits the movement of the animals. You can imagine that if I have now another animal here, 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 I recover this picture with the wolves that we were seeing in the first video. That's the idea. So then the question, how does this additional spatial scale influences the encounters between animals? And I'm not going to do the whole math. I just want you to get a clear idea of intuition of the directions I'm taking with this approach. You can see, as I was saying, that if I have two animals and I place their territories in the way that I parameterize the model, then in this case, for instance, they are very unlikely to find each other. See that these 95% contours, they don't overlap. So it's very unlikely that they find each other, right? In this case, on the other hand, because they are always, they can be all over the place, then the probabilities are going to be definitely much higher. So you can already see that this definition, I mean, this new spatial scale is actually very, very, very important. And now that I have my models well defined and I have them ready to do my calculations, let's get a very brief intuition of how I'm going to define the encounter, which I think is going to be very easy for all of us, but it's going to allow us to define a key property of this problem. So I'm going to think of a prey and a predator. A prey is a poor animal that is eaten by a predator, so zebras and lions, for instance. And what I'm going to say is, well, they are living all over the place. I have my predator, and the predator has some region around it that will allow her to eat wherever it finds inside this region. So here, for instance, the prey is safe because it's outside the red region, but at some point they will get very close, and then the lion is going to attack the zebra. That's the idea. So the key property to be able to calculate these encounters is this, the key variable, sorry, is this R, is the distance between individuals. So it's not important if my animal is here or is there, the important thing is how far they are from each other. So the key thing to obtain these encounter rates is to obtain this distribution for the distance and then do some mathematics with that distribution. Again, I'm not going to do the math because it's a lot of integrals. You can go to the references, we can discuss them at the end on the Slack or wherever. I would be very, very happy to do so, but let's keep the story moving. Let's not distract ourselves with math. So again, I can do this calculation in the two scenarios that I defined. I'm going to put you now the expressions of the encounter rates. They are kind of ugly. One of them is uglier than the other, as you can imagine, but we're going to discuss some important features of them. We're not going to go into the math. So this is what I get for my harmonic oscillator model, for the Austin-Uhlenberg model. And this is what I get for the ideal gas model. So don't worry, I'm going to explain the parameters very carefully. So in this case here, this gamma is basically giving me how good animals are attacking. So you can take it equal to 1 and forget about the thing. This sigma R here is basically the variance of the distribution of the distances between individuals, right? So as I was saying, all the important information is in this distribution for the distance between individuals. So my encounter rate here is going to depend on that, on the properties of that distribution. Whereas here, nicely, my encounter rate only depends on the size of the arena in which I put my ideal gas particles. The rest is not important at all. And this is, of course, provided that I take Q, the limit in which Q goes to 0. Okay, so I define that Q for mathematical feasibility, and then I take the limit Q equal to 0 to keep things easy. So what is important about here? Again, we can discuss the math later, but what is really important about here is something that is going to blow up now very easily. In this case, in the case of the reflecting Brownian motion, I only have dependence on one spatial scale, which is the size of the Earth, if you want, of this disk. But here, I actually have 3 spatial scales that are determining the probabilities of encountering. One is the contour of the 95% region of one individual, of the other individual, and the distance between the centers of the encounter area, of the home ranges, as we define it. It's very easy to see that in this figure that we had before. In this case, the only important thing is my capital R. If my capital R is larger, the animals have more territory to move around and they will be less likely encounter with each other. Here is not only that, what is important. Here it is also how close the territories are, how large each one is with respect to the other. And I have a huge, 3, which n larger than 2 many times is huge. I have 3 scales to play with that will determine my, my encounters. And that's the key difference between simplified models and not so simplified models. So just to give you very, very brief intuition on how can we compare this. Imagine in this setup here, in which I'm taking rho 2 or rho the predator to be equal to the capital R. So basically my territory of one animal is equal to this hard disk. And then I have the territory of the other guy that is smaller and inside the other one. And the question I'm going to make is, okay, what happens if I shift this territory, this blue territory, around the territory of the other animal? So basically, if I say, first my animal, my blue guy, my prey, is going to spend most of the time in the center of the territory of the predator. Now, the animal is going to shift her territory and is going to put it right here. Now, a little bit more farther, and now a little bit more farther. I'm going to take these 4 points. And the question that I'm going to make is, how does each of these cases compared to the case in which my animals are moving equally likely all over the place? That's my question. I think it's easier if we see it in 1D, although 1D many times doesn't work in ecology. It helps us to make an idea of what's going on. So the first case I showed is this one, where I have my Gaussian for the gray guy here and the Gaussian for the blue guy here. And I'm going to shift, my Gaussian to reduce the overlap, basically, and calculate these encounters. And I'm going to compare each of these scenarios with the case in which all of them are uniform inside the territory. So these are the two scenarios that I'm going to compare. Let's see what happens. Well, what I see is, because I'm keeping this capital R constant, the case of the reflective Brownian motion is constant too, because it only depends on capital R, and capital R is not changed. It's always the same. But here I already have an interesting result. The important result here is that the Orsten-Gulenberg model actually sometimes is going to give me higher encounters, because in this case, the animals are going to be most of the times closer than in this case. But as I move around the territories, this is going to decrease, 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 and eventually I'm going to be below the reflective Brownian motion result. And I can do this for a huge bunch of possibilities. I can combine the way my territories move. I can play with my spatial scales. And what I get, if I do this kind of more systematic review of the parameter space, is this figure that you can see here. So what does this figure mean? Because it has a lot of information. Here I'm representing the ratio between the encounter rate for the OU model. So the ratio between this black curve and the red curve. I'm condensing the information in this ratio. Whenever I have red, it means that OU is larger than the reflective Brownian motion. And whenever I have blue, it means that it is smaller. And what I get is this picture that you see here. So as you can see, well, it's kind of nice, has very pretty colors. But what is important about this? The important thing about this is that the only region in which this ratio is equal to 1, which is where my reflective Brownian motion would kind of give me a good approximation to the more elaborated model, is this very, 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 very tiny region of the parameter space. In all the other places, my encounter rates can be higher or lower as I remove details of the microscopic model of my animals. And this is very important. It is very important because I cannot tell you, okay, whenever you use your logistic equation, you will always be over calculating encounters, or you will be always under calculating encounters. No, it's going to depend completely on the context of the problem. And that's an additional complication to my study, because I have to really understand the context. It's not only, okay, you almost get like, I don't know, 20% overestimation, you see you are in this. No, here it's like almost negligible. Here is 3 times higher. And if I move the parameters, I can get completely different things. So this is a crazy way of saying that things have to be thought more carefully. And at least we have to think if the system we are working with lies within this region. So for the case of the yeast that I showed you in the beginning, we could kind of say, yeah, fair enough. They are around here. For the case of the wolves, maybe we are here. Oh, sorry, we are here. Oh, I don't know where we will be. I have to think. I think it's around here. Anyway, what is the important thing? The important thing is that the frequency at which animals change is going to depend on the movement, on the model that we use for the movement, and on the details of the movement. But there is another important thing, right? It's not only important when they find each other, but also where they find each other, because that's going to give me more information. And I guess at this point, your intuition, if it's working, is going to have a pretty accurate answer to this question. If my animals have a limited overlap between their territories, then I should expect that they would encounter most of the times over here, right? It doesn't make a lot of sense that if the blue animal moves around here, we'll find the gray here. They will kind of find each other in the borders of the territory. And using theory from stochastic processes, if we assume some properties for this movement of the animals, that sometimes they work, sometimes they don't. So here I'm assuming that individual 1 moves regardless of how individual 2 moves, which is a big assumption, but let's move from here. Then I can calculate the probability of the animals to find each other in every point of the space. I can find that. I can calculate that. and I can give an estimation for that region. So here what I'm showing you in this picture on the right is a number of encounters. I think it's eight encounters between the gray and the blue guy. And on top of that, I'm giving you the 95%, the region of the space in which I will find 95% of my encounters. And it's limited by this black region. So as you can see, it works pretty well. I mean, the gray curve has no information about data. The gray curve is theory. The only thing is, of course, the parameters are the same. Okay, so now we know, and again, sorry, if I don't assume this movement, then my encounters can happen everywhere in the space with the same probability, because animals are everywhere. And in principle, this a more realistic description of movement is limiting where animals can find each other. Okay, nice. So far we have kind of, I think, a fair answer to this question of how physics can help biology. But we said that this was going to be a two-way relationship in which physics helps biology, biology helps physics. So let's see how can we address the other question. Let's see how much biology can we learn from this. And once we know how much biology we can learn, we can develop new questions. And the new biology that we learn is going to challenge us to develop new physics. That's the idea. And to show you how much ecology we can learn from this, I'm going to switch a little bit from integrals, distributions, SDEs. And because it's Friday, I'm going to switch from wolves, which are very nice animals. I'm going to switch to these very small and tiny monkeys. So these tiny monkeys are white macaques. They live, the population we work with live in Panama. And I'm going to show you a movie of how these animals move. And the data that I'm going to show you is not ours, it's by Grace Davis and Megan Cruford, who are in the United States and also work in Panama and in Germany. And what they did was to track these monkeys, as we did, as I told you with the wolves, and they collected data on the position of these monkeys for some time. I think it's 3 years. About these monkeys, if you want to know a little bit more, they live in groups, and within the groups they are very friendly with each other, but they really don't like, again, finding guys from other groups. It's not a very good idea that one monkey from these groups finds the other one. So again, this is Panama. This is a research station in Barro Colorado, which is an island that has been used for a lot of ecological studies. And let's see what these monkeys are going to tell us. Well, again, as you can imagine, each color represents a monkey. Here we have 7 monkeys. I'm not going to use these 2. I'm only going to use these 5. And you can see that this is the territory that they use. Each point is a monkey. Sorry, because the video gets stuck a little bit. But basically, the monkeys are moving around whenever they want. Yeah, they don't seem, again, to have a lot of overlap with other guys. They occupy their territories. They don't go into much trouble, although here there is some trouble between these 2 guys. Well, this is the way these animals find each other. So, move around. So looking at this data, we can already, if we are interested in saying, okay, where are these animals going to find each other? Where these encounters are going to take place? Well, I think it's pretty fair. Oh, sorry, what happened? I would say it's pretty fair to say that most of my encounters will concentrate around here. At least by intuition, I would say that, right? I would say, well, yeah, they live around here. So I can calculate my distribution of the probability of finding an encounter, and I can represent it on top of this plot, and I can see what happens. And this is what we did in this study here that was actually led by Michael Nuna, who is now a professor in Canada, in the University of British Columbia. What he did was to develop a statistical tool that allowed him to quantify from this data, from the position of the animals, allow him to quantify the theoretical calculations that we develop. So basically, we calculate this probability distribution analytically with doing math, and then he does some statistics and he develops a statistical estimator for this tool. And I have to say that he developed that with Chris Fleming, who is a researcher in the Smithsonian Institution as well. So as you can see, when we plot the 95% contours of this distribution, they follow very nicely the borders between the territories, which is something that we would expect. And working with properties of this distribution, we can actually give intuition on which are the actual frontiers between these territories. So this is kind of using the data, the way animals move, to see how are their borders, to see, as we do with countries, in the case of Spain and France, we have the Pyrenees. Here there is nothing. So what are these frontiers that actually limit the territory of each animal, as politicians do with the territories that limit each country? It's the same, but we are using data and we are using these models. Okay, so now we have these regions defined. We know that from these animals, from the position of these animals, they will encounter within this region. So if you remember, I told you that animals, I mean, these monkeys, they really don't like so much finding guys from the other group. They are not very friendly with animals from the other group. So the fact that encounters here are more likely than here, for instance, I think, or I want to make that point, makes this region of the space different from this region of the space, right? It's not the same, the way people behave when they are in a place where they can find some danger than the way they behave where they cannot find any danger. So it's the same with animals. So now the question is, how can we use this, which is something that we obtain from the models? This estimation is given by the models, by a definition, by an integral that we solve. Can I use that to learn how these animals behave? And the question is yes, I can. So what I'm showing you here, I'm sorry because this is a lot of information altogether, but we are going to grasp it step by step. What I'm showing you here is the mean speed at which animals move. This is the x-axis, and the y-axis is different animals. So the mean way animals move when they are in their own territory or when they are in the red green. Okay, so for instance, imagine that, of course, each of these ticks represents one of these 5 monkeys, and I'm going to tell you, okay, how does this monkey move when it is inside this region or when it is outside the region? And as you can see, if I take the average over the 5 monkeys, it's pretty clear, it's statistically significant that the animals, when they are inside the red region, move faster than when they are outside the region. And this is not only true for the whole population, it's also true for almost all individuals except for 2 guys. Okay, so this is the point. The point I want to make is that doing math, using physics, using models from statistical physics, we define this region of the space. And then we could look how different our data is in different regions of the space. But just because we had the theoretical support that was defining the region where we had to look at. If not, it would be pretty hard. It would be harder to say, okay, where I start from. So this is the key contribution of this relationship between ecology and physics, at least in this project. So this is what I wanted to tell you about this specific work. I want you to finish with some other things we are working on, because as Raul said in the beginning, I've been here for a year. So we are very few people so far, and I would love to have more people working with us. So if you are interested in any of these projects, we can talk, and it would be very great to see if you are interested. So I want to give you information on the things that people are working on right now, we are working on, and how happy we are with how things are going. So the first thing I want to discuss, or this is going to be very brief, it's how the landscape is going to affect all these results. So far, as you maybe are thinking or wondering, all my animals, they don't look at the space. My equations don't have any information about the space. And it is not the same for a monkey to live, or for a wolf to live in one type of landscape or in the other. Or in the case of jaguars, which is the animal with which Gabriel is working. Gabriel is a postdoc at ICTP. It's not the same living in the forest than living in an open space, or living close or being close to a river than far away. And this equation, that is the refined model we discussed, has no information about that. So something that he's working out is how these landscape heterogeneities are going to affect these results. And this is work also in collaboration with Ronaldo, who is a researcher in the Instituto Chico Mendes de Conservación de Biodiversidad. So some preliminary ideas that I can give you is that, are from this movie. So here, this dark gray line is a river, and this blue point is a jaguar. You will see over time how it moves, and we'll discuss what's going on here. So at some point, yeah, it starts moving all over the place. I have to say that these trajectories that close the river is because of the discretization of the trajectory. The animal is probably doing something around here. It's not swimming all the way. But anyway, the animal is jumping from place to place with different rates, boom, boom, back, forth, and it's doing kind of nice movement. So this is just a sample trajectory, one animal, not a lot of statistics to do here, but you can see many things. So I would say the first thing is that the animal goes a lot to the river, so something is going on there, definitely not the same as this yellow region here. And also another interesting thing that Gabriel also pointed out is that the animal has here a core of locations. It's very frequently going here. And this is something that is characteristic from this model, right? Remember that they were Gaussian distributions. So probably for this model, maybe if we want to parameterize this animal to this model, lambda would be around here. This would be the center of the harmonic potential. Anyway, this is something we are working on. Well, Gabriel is leading all this, and hopefully we will get very nice things soon. Other things that are interesting to understand is, well, so far we are characterizing frequency of encounters and location of the encounters, but sometimes the important thing is not how many times the animal encounter in 2 hours or where are my encounters going to happen. Sometimes it's more important to say where are animals or when an animal is going to encounter for the first time. For instance, imagine that, I don't know, 2 animals want to reproduce and they have a breeding season. They have to mate in a month. So then the question is, will animals encounter each other in that month? After that month, I don't care. I don't care if after that month they encounter 1,000,000 times. If they don't encounter in that month, they are screwed. Another question that is ecologically relevant in this context, what is the probability that a predator encounters a prey within the next, I don't know, 3 days, 2 days, before it dies of starvation? No? They have a limited time to eat. If not, after that, the model can predict, again, that they will find 2 million of zebras. If they don't find one in 4 days, they are done. Another question in which these metrics are important is, what is the, well, this is the first time, the one I discussed before. What is the probability that a female and a male encounter with each other within a mating season, for instance? So to approach all these questions, we need to use what is called first encounter statistics, which I can tell you is way more complicated than the stationary state statistics that I discussed here. And it's something that Benjamin is working on for his master work. And to give you some idea, I think with pictures, at least I understand things better with pictures. If I will go back to this representation of two animals moving and all the encounters that they make, we said that the region of the encounters will be over here, that with some frequency they will start encountering. I could give you a probability periodic time of these encounters to happen. What with Benjamin is trying to obtain, what he's working on, is a much more complicated question. It's a much more challenging question, which is, out of these 8 encounters, which is the first one? What is the probability that my first encounter is this, that, when that encounter is going to happen and where it's going to happen? And that's a very, very hard question to address. So this is it. This is all I wanted to show you. I just want to finish as always, giving thanks to, first again, to the organizers. This was a very, very good and very important event for everyone. And also I want to give thanks to everyone that has helped me in all these projects, and not specifically in this project, but also in the way that we are developing things here. So Corina, who was my postdoc mentor in Princeton, Cristobal, who was my PhD supervisor, Justin Calabrese, which is actually the leader of the senior researcher leading all these ideas of encounter projects. He's the principal investigator of this project. Simon Levine, who was another postdoc mentor I had in Princeton, and Juan Bonachella, who is a long-time collaborator and friend. And more importantly, I'm sorry to put these guys down, although especially because this will probably go to YouTube, but anyway, especially thanks to people that are actually now getting all these nice things done, which are Vivian, Benjamin, and Gabriel, who are working on this, and well, of course, many other people that have worked, and hopefully in the future, some of your faces will be around here. And also thank you to the people that pay for our research, which is FAPESPI, Instituto Sapieira, and the Simons Foundation. And I'm happy to take questions.
So thank you very much for your talk, Professor. I think everyone enjoyed a lot. And we already have some questions in the chat. So please feel free to ask your questions to Professor Ricardo. You can make them in English or in Portuguese. You can read to him. And Gabriel, I think that Gabriel Soares, you have the first question. You can open your mic and answer and make the question.
Obrigado, Raul. Acho que vou fazer em português mesmo a pergunta, se não for muito incômodo.
Se fala despacio, se não entendo, pergunto eu para você.
Tá bom. Então, minha pergunta é mais sobre, já que você falou de efeitos do landscape, né, do território, né? Eu estava pensando em como, se você tem alguma pesquisa voltada a como perturbações no clima ou perturbações tipo queimadas e tal, como que se tem alguma pesquisa voltada a esse tipo de efeito do clima nessa territorialidade dos animais.
Então, vou tentar responder em português. Se a coisa não fica muito boa, mudamos para inglês, mas eu acho que vai dar. Estou praticando muito na quarentena. Então, a coisa, nós não, neste contexto de modelagem de movimentação de animais, não temos consideração do clima, porque a gente agora está trabalhando numa escala temporal que é muito mais curta do que as mudanças climáticas podem acontecer. Então, na nossa pesquisa agora não temos essa pergunta, mas nós temos outra linha de pesquisa que não discutimos agora, mas fico feliz de discutir se vocês quiserem no Slack ou no e-mail ou qualquer coisa, que é a formação de estruturas espaciais de vegetação. Então, nós o que estudamos é como é que as plantas interagem com elas, umas com outras, para elas formar esses padrões espaciais que nós observamos. Lá, nesses modelos, nós sim que temos em conta o clima, as mudanças na precipitação, como é que os ecossistemas vão responder a essas mudanças, essas coisas. Nós sim que estudamos, mas num contexto de vegetação. Aqui na movimentação de animais, não, porque tem pessoas que fazem essa pesquisa, mas no nosso estudo nós temos a nossa motivação escalas muito mais curtas do que as mudanças climáticas, e como é que elas encontram os dados que nós temos 3 anos, 2 anos, 1 ano. Então, nós agora não temos essas perguntas, mas tem pessoas que sim, por exemplo, tipo no caso do jaguar, do jaguar aqui, da onça, desculpa, a onça. O Gabriel vai me matar. A onça aqui, já tem algumas coisas para discutir com a mudança climática, não? Por exemplo, se ele gosta tanto dos rios, da cobertura florestal, como é que se a floresta vai extinguindo, como é que a movimentação vai mudar, ou uma pergunta que o Vítor, que ele acho que ele está por lá, que trabalha com Roberto Krankel, tem, como é que se a floresta é fragmentada, é quebrada em troços pequenos, como é que essas populações vão mudar. Então, tem pessoas que sim que fazem essa pesquisa. Nós, por lo de agora, mesmo pode ser que esse projeto volte um pouco para lá, mas por lo de agora não trabalhamos nisso. Mas eu posso compartilhar com você referências, se você tem interesse nesse tópico.
Tudo bem, obrigado, professor.
Eu agradeço.
Agora, eu acho que a Thaís Cristina. Thaís, pode fazer a sua pergunta.
Raul, ela pediu para você ler.
Okay. So, in these points where they encounter each other after action of the predators, is there a drastic decrease in the population or it is balanced?
Well, that's a very, very good question. Yes, that's something. So for us, for instance, we are using this prey-predator motivation, but it's very clear, no, that it doesn't make a lot of sense to think, okay, my lion is going to encounter the zebra 4 times because the first time is going to kill her, and then there is no point about. And that connects, for instance, with the problem that Benjamin is working with. It's only the first, the one that interests us. And I think what actually Thijs wanted, I mean, I don't know if it's what you wanted to ask, but something that is very important is how the other preys are going to react to that encounter. No, if I have 8 zebras, then the encounter itself is going to affect the population, and they are going to spread out. That's something that we are not considering. We are, again, as I said, is n equal to 2. But that's of course something that it's important to take into account if you are interested in short time scales and short time encounters. It's a very important question, but we are not, I mean, in this project we didn't investigate it, and we are not working on it so far, so I cannot even tell you. Of course it's going to decrease, I don't know, especially imagine if the prey is social, they live 20 preys together, and you have a lion attacking, I'm pretty sure they will disperse. Probably eventually they will come back together, because if they are social, they really need each other. But this fission-fusion dynamics is going to be important. But unfortunately, I cannot tell you any spoiler on that.
So I think we have time for one more question. Carlson, you can make your question.
Hi, can you guys hear me?
Yes, perfectly.
Well, thank you for your presentation. So I have a curiosity. I'm not expert in this field, so I try to explain my curiosity. So in your calculations about encounter rates, do you know? Did you consider the number of preys which attacked by one predator? Did you consider this? Do you know? I don't know if this my curiosity is clear. So, did you consider the number of preys attacked by just one predator?
Yes. So yeah, the question is perfectly clear, and it's a very good one actually. So again, this is n equal to 2, so there is only one prey that can be attacked. So this being said, in our calculations, remember that we have 2 guys, and I'm saying prey and predator because I don't know, it's a use of words. You can imagine a mosquito and a person, or two animals competing for food. It's only encounter between trajectories, if you want to take into a more abstract way. Of course, when you introduce more animals, then it makes your point is a very important one, because the number of, so how can I explain this? In the sense of having food, it is not the same in terms of the dynamics of the population to always go eat in the same place, or to be able to eat from different places. If you are consistently, for instance, attacking the same group of prey, that group of prey will probably move, right? Imagine, I don't know, if a wolf wants to hunt in the same tree, after 4 attacks, animals are not stupid. They will leave and they will find another tree, and that's it. If you jump from tree to tree, maybe it's a smarter exploitation of the resources. So calculating this number of preys is a very important metrics too. We didn't calculate it here, but yes, it's very important. We are not considering, I mean, again, no spoiler on that. I think it's very related to Ty's question too, but no spoiler on that either, sorry. But if you want to calculate it, just tell me.
Okay, so now I think that it's time for our picture. I would like to invite Professor Ricardo to stay with us so we can take a nice picture.